We didn't have these images yesterday. Apparently all it took was plugging in a cord that had fallen behind the box. But I never said I knew my computers very well. So I wanted to go back through them and just do kind of a review of the material that I talked about that wasn't necessarily shown very well without the images. Let me see my... Alright, so we started off with occipital lobe, and the next slide shows the same diagram we were talking about in the thalamus. I followed through the pathway, and we'll do this again and again when we get to the eyeball. But the bipolar neurons, the first neurons, would be located in the retina, and then they would synapse with the second neurons, some of which cross and end up in the thalamus. Okay? So part of the thalamus is, one of its major functions is a sensory relay. And so for light action impulses coming in, the nucleus indicated here is the lateral geniculate nucleus of the thalamus. And I'll show that to you on the model later on the slides. And then the third neuron comes to that primary auditory cortex. And processing the firing of those cell bodies that form the gray matter of that primary visual cortex is going to create the image. All right, and recreate what it is that we see out in front of us. So it shows you both function of thalamus and the region of that visual cortex. Interestingly enough, the cortex is broken up into different components. So as something moves across your field of vision, you detect that as it crosses from one side to another. That's picked up as movement. The shape, various forms and colors are picked up in different areas. So it's not just oriented to certain signals. And so here's the fibers coming in from the thalamus. Okay, temporal lobe. We had the primary auditory cortex here. Taste on the inside of that, and sensory speech up in this region. And also associated with the temporal lobe, I wanted you to remember the hippocampus. And so here is the lower region of the hippocampus. This is the dentate gyrus that I said you didn't have to worry about. And the hippocampus itself is that coil of gray matter in this area. So this is the inferior horn of the lateral ventricle, and its floor is formed by the hippocampus. Okay? So we're going to go through a lot of these structures, the structures of the cerebral basal nuclei as well, as we go through those structures again today. And then in lab, I'm going to show some slides of the coronal sections and review those as well so that those make sense to you from the images that you see. Okay, so parietal lobe, the only thing you need to be able to identify in lab from lab is the postcentral gyrus. So in our lab quizzes and lab exams for the occipital lobe, you don't have to be able to identify this precise primary visual cortex, just the occipital lobe. Okay, and you don't have to be able to identify the primary auditory cortex, but I do want you to identify be able to identify the hippocampus. All right, so knowing what lobes those other functional structures are in is a lecture concept. Okay. This was the sensory homunculus that I kind of showed to you on the board, and again, kind of the knee at the top of the longitudinal fissure with the foot hanging down within the longitudinal fissure. And then the person is kind of like hanging, well, we have knee, foot, and hand, and then face along the side. I'm not going to test you on this in lecture or lab. If I put a point here and have you tell me if it's a finger or a lip or a whatever, just recognize foot is this way, hand is this way, and knees kind of the top there, the longitudinal fissure corner, and that's about it. Okay, I'm not going to have you recognize all the other structures. Frontal lobe, only thing lab-wise you need to recognize for frontal lobe is the precentral gyrus. All right, and it has its own motor homunculus with the same sensory and motor areas are on different gyri, but about the same approximate location. And just showing you where these numbers come from for specific components. You don't have to memorize these numbers at all. It's just kind of showing what we do with those areas. Okay. All right, so insula. Remember that was what we see, the cortical area that we see if we open up the lateral sulcus, and here it's actually been cut out, but it's a continuation of the surfaces of the brain. And then this slide, if you can't remember what I talked about the insula, we had it was missing in our lecture notes. So the insula lights up when people crave drugs, anticipate pain, empathize with others, listen to jokes, see disgust on someone's face, go to a party and nobody pays any attention to you, insula is activated. Try to do, the rotten food versus the good food. In fact, there's some people that have disorders of the insula that all they'll eat is rotten food. All right, so we get different behavioral problems with this. There are certain people that you may have met or seen in movies that they just don't seem to have any social skills. They can't tell when somebody's mad at them. They just keep saying stuff that makes people madder and madder. That involves the insula, okay, when they can't. Part of it are other structures of the limbic system we'll talk about today that involves reading facial expressions from someone else.
From somebody who had like Asperger's, would they have a problem with their insula?
A lot of Asperger's involves a lot of the cognitive effects. That's a form of autism. All right, speech and language centers. Remember we have the sensory speech area involving both parietal and temporal lobes, and then the motor speech area involving how you actually form a sound rather than interpretation. So we talked about speech. And then we moved into the basal nuclei, which was the more difficult area to understand without the slides. So don't want that one. You recognize some of the diagrams I was trying to draw on the board. So basal or cerebral nuclei, here is the caudate nucleus with the putamen, the globus pallidus, and thalamus in the center. So this is starting in the frontal lobe, moving into the parietal lobe, and ending up in the temporal lobe. All right. And again, when we do the clay modeling, by the way, you need your clay for Tuesday, and then gloves, closed-toed shoes, goggles for Thursday. All right, so I'll remind you about that part of it on Tuesday. This might be an easier view to see the contrast. So the thalamus is here. Remember that on the other side of the thalamus is going to be the third ventricle. And then this dotted line here shows where the head of the caudate would extend to. Here's the putamen, and here's the globus pallidus on this portion here. So the more anterior structure, when we cut through the head of the caudate, and I'll show you this on a Netter's diagram, we're not in the third ventricle. Okay? By the time we get to the thalamus, we're cutting through the body, the putamen, and the globus pallidus, and we're in the inferior horn cutting through the tail. So it really helps to start visualizing now, if I can see this, where am I? And the third ventricle is a really good locator. So again, head of the caudate, body and tail, thalamus, and the putamen and globus pallidus would be between the blue and the red regions. So that you have this diagram on your lecture notes, and there's a really complicated circuit between the structures of the basal nuclei. So we have case studies on both of these in both sections, and they may go into it more detail in their paper and or their PowerPoint. But essentially, remember I said the basic function of the basal nuclei or cerebral nuclei is to do what? Suppress unwanted motor movement. And that loss of function can be seen as both tremors, which makes sense, that's unwanted motor movement, but also stiffness. And the stiffness comes from too much flexor and too much extensor control. Therefore, you can't move the joint. So the muscles are trying to flex, there are also other muscles are trying to extend the joint, and you result with a stiffness at the joint, and nothing moves. So it seems contradictory to have stiffness as unwanted motor movement, but it's excitation of both sets.
So like cerebral palsy, when they get like stiff, arm movements and stuff, is that?
Cerebral palsy can be due to a variety of things because it's caused by a lack of oxygen during the birthing process itself. And that could be basal nuclei, it could also be frontal cortex. So it varies on the person. But certainly this can be involved as well. So this top diagram is a little bit more simplistic. It shows the basal nuclei. Ganglia, again, is an old name and it's not appropriate because they're nuclei, not ganglia. Cerebellum, and we'll talk about the function of the cerebellum on Tuesday, but the cerebellum is going to activate the cerebral cortex. All right, so cerebellum is coordinating events and movements. So as I reach out to get something, my joint sense, my visual input of where that structure is, and my inner ear balance is all telling the cerebellum where my body is in space. And then my cerebral cortex is telling my cerebellum, this is what the movement is going to be. I'm going to move my arm out here. These are the muscles that I'm activating. And the cerebellum responds to that by changing the activities of the cerebral cortex. So maybe I'm moving up this way, and the cerebellum knows I want to go this way. So it will send a signal back up to the cerebral cortex to adjust my hand very smoothly and bring it back to the site that my eyes are saying I want to go to. All right? So as we'll see on Tuesday, if the cerebellum isn't working, we have these wide swings that get worse and worse and worse the more you try to focus in on a movement. So the cerebellum is being told from the cortex what it's going to do, and it responds to that to change the movements of the cortex. So it's activating the precentral gyrus. All right? The basal nuclei are inhibiting that. And the result that ends up is the net outcome. So whatever is left over from the active versus the inhibitory influence is what you actually end up doing. This lower diagram that you also have in your lecture notes shows more of the relationship. It doesn't have the pluses and negatives, but it kind of shows the substantia nigra is that black stuff, again, down in the midbrain. And that's going to send information to the caudate and putamen components. And the lower portion of the thalamus communicates with the lobus pallidus. Those two communicate with each other, and then there's this reverberating circuit between the thalamus and cortex, which is also getting information from the cerebellum, and we end up with a movement or not a movement. Okay? And that's the level that you need to understand. You don't have to know which one is more positive or negative or the neurotransmitters between them. So here's the cerebellum. All right? Here is the substantia nigra in the midbrain. We'll talk about those structures on Tuesday. A small portion of the thalamus is involved with this, and then the rest. So it's information going back and forth and into the cortex as well, that we end up with our controlled movement. Okay, so looking at this structure, again, remember we're seeing the third ventricle. So we're going to be back about halfway, a little more than halfway, probably in the parietal lobe. So instead of the head of the caudate, we're seeing the body of the caudate along the lateral ventricle. Here's our third ventricle. So the structure of the brain forming the wall. When we touch the floor of the third ventricle, when we touch the side of the third ventricle, we're actually touching the thalamus. Okay, so we're always going to see thalamus when we see third ventricle. These are projection fibers here, and then we see the putamen and lobus pallidus. Here we're seeing the inferior horn, so we're seeing the tail of the caudate nucleus coming around, and then the floor of the inferior horn, the lateral ventricle, we're seeing the hippocampus. So we'll look at these structures in lab as photographs of the coronal sections. We haven't just started talking about the fornix, so I'm going to wait till we get to the hypothalamus today and talk about those structures. Here again, caudate, thalamus, third ventricle, hypothalamus in this region. Now this is from Netters. So again, we're seeing the caudate nucleus. Coming back here, the tail is going to be in this area here in the roof because this is the hippocampus. Right here in the floor of the lateral ventricle, forming the fornix going forward that will pick up with the hypothalamus. And then the putamen and lobus pallidus are going to be in this area right here. kind of been cut away. Okay, so that's the head of the caudate that you're seeing there. And in some of our models, you can see that. So we could, if we pull this up like this is a membrane separating the lateral ventricle. If we pull that up in the sheep brain, we have to cut that septum pellucidum. If we pull it up, we're looking into the lateral ventricle. And that structure that you're seeing right there, bulging in to the anterior horn, is the head of the caudate. If we take this off, then you're seeing a view a little bit closer to that. So here is the head of the caudate. This structure would be the insula. Putamen and globus pallidus are going to be between those two structures. Okay, so underneath the insula, would be the putamen and globus pallidus. And this model, when we do lab, you can see that. In fact, let me take this off now. So here's the insula. If I pull this off, that would be the putamen, just deep to the insula. On this surface of it would be the globus pallidus. You can kind of see the triangle shape of it if you look at it in lab. And then here is the lateral ventricle. Remember, recognize the shape of the lateral ventricle. And what's bulging in right there is this structure right here, which is the caudate nucleus. Okay? So the head of the caudate bulges into the lateral ventricle. So again, we'll have opportunity in several labs to repeat that illustration over and over. Now this is a transverse section. Top of the brain has been taken off. All right? And it's the same view as this, except we've cut through it here. So it's still looking down on top of the brain. You can get this off of the D2L PowerPoints for lecture, or if you have netters, I think it's plate 104. And this is really helpful when you're looking at the coronal sections. Because if you're at the very front, you're not even cutting through the anterior horn of the lateral ventricle. If you're cutting through the head of the caudate right here, you're cutting through the lateral horn, you're seeing the head of the caudate, but you're not seeing the thalamus or the putamen, because we're not to those points yet. We're just right at this level. When we're cutting through the third ventricle, we can see the third ventricle, we can see the thalamus, we can see globus pallidus and putamen. So it helps you see where you are as you move anterior or posterior in those coronal sections. Okay, so it's a good one just to have on the side there to give you an orientation. And this is the same structure, just as a smaller view. All right, so you also have this material in your lecture notes. And again, it's just kind of another circuitry of the basal nuclei. For interest's sake, I'm not expecting you to reproduce this, even for extra credit, on the lecture exam. Okay? The extra credit part for our lecture exam is one of the pathways that we'll be describing at the end of next week. So I do want you to understand that all of these are components of the basal nuclei, but even specifically one of these functions with this specific structure, I'm not going to ask. I certainly want you to know that these functions are involving the basal nuclei, but you don't have to associate specifically to the caudate or specifically to the globus pallidus. Okay? But just as we go over it, you'll understand its components. So when we have a lesion of the basal nuclei, these are the most common forms of demonstrations. And the choreiform movements are the dance like Huntington's chorea, is a genetic lesion of the basal nuclei. The athetoid movements were those kind of movements that I was showing like this. It was just a constant, movement like that. And then Parkinson's disease is the stiffness of the gait. The hemiballismus is a flinging motion. So it'll just be a kind of a throwing of the arm rather than the sinuous dance-like forms. All right, then we ended up lecture yesterday by looking at the thalamus. And the thalamus is seen as this central region right here, but it also extends more posteriorly back here as the pulvinar region of the thalamus. So if you look at this model, this is a brainstem model that we have. This is a ventral view. This would be insula right here. And this is the optic tract leading back to the lateral geniculate nucleus. And then here is the posterior aspect of the thalamus, which we don't see on a sagittal view, called the pulvinar. Okay? You don't have to identify ventral medial nucleus or anterior nucleus, just the lateral medial geniculate and pulvinar components of the thalamus. So it's this model, and it's to see the pulvinar, you're going to look at this view right here. So my two fingers are on the pulvinar structures of the thalamus. Posterior structure. All right, remember the thalamus functions primarily as a sensory relay, so most of the gray matter is formed of these cell bodies right here. They're the third in a sequence of sensory neurons extending out to the precentral cortex, if it's pain, touch, and temperature, extending to the primary visual cortex if it's vision, extending to the primary auditory cortex if it's hearing. All right, so it's going to go to any of those depending on the type of sensory input it is. And then this is new material. All right? So hypothalamus